just takes a little moment. Yeah. Okay. Yeah, so that's done. Okay, so I think we should start. So it's a pleasure to have Andy Dobson here today, and he will talk about a completely different aspect, I think, of the COVID-19 pandemic. And well, I think I would like to give the word to you, and maybe you can say a few words about the background and how you came up with this idea of research.
Okay. Hi. Thank you so much for inviting me. It's nice to be here, but I really wish I was there. I could have gone to watch Barcelona play Espanyol last night, or I couldn't have gone. If I could have been there, I could have gone and seen it. But yeah, it's a shame not to all be together. What I'm going to talk about relates to COVID, and I'll keep returning to COVID through the course of the talk. Lots of what I'm going to actually describe comes out of a set of working groups we had based initially at NCES, the National Center for Ecological Analysis and Synthesis in Santa Barbara, and then at a place called CICIG, which is for the synthesis of data, ecological data and environmental data relevant to human populations, which is in Annapolis in the eastern US, and it desperately likes to think it's like Santa Barbara, but it's really very different. They're both very nice places. What those working groups were looking at were, what are the conditions, the mathematical conditions and the ecological conditions that lead to diseases jumping from their natural reservoirs into humans. So very much, I've titled the lecture Emerging Diseases, Causes, Mechanisms, and Prospects. And I can actually have it in Spanish, but I'm sorry, this will be the only actual piece that appears in Spanish. So I will be trying to describe some mathematics for the mechanisms that cause these pathogens to jump across, looking at COVID, and then a more brief final section deals with what is the cost of stopping this happening again, and how does that compare to the cost that we're currently having to pay for having let it happen this time. Partly to convince you, this is not the first time this has happened, and it's definitely not the last time. This is the most expensive it's been, both in terms of finances lost, jobs lost, and very much human lives lost. So we want to know how to stop it happening again. Okay, so let's jump in. This is where we first got indications that COVID was out there. This is the wet market in Wuhan, where many people go to get their food. In many ways, it's equivalent to the beautiful markets you have in Barcelona. People go there to get their meat. They don't have as wonderful cheeses in China as you do in Barcelona, but they get their fruit and vegetables. A tiny proportion of the market also deals in wild animal species. And it's deeply suspected that it was from one of these wild animal species kept live and then slaughtered in the market for people to take home fresh that we think Wuhan initially came from. We're now getting evidence it might have been circulating a little earlier than it was first detected in this market, but literally at the end of last year, this is from the 30th of December, those of us who are nerds and interested in these diseases follow this thing called ProMed that sends us out information 2 or 3 times a week of unusual disease events. And this came out 30th of December to say people were picking up an unusual disease event around this wet food market in Wuhan. And people had this strange pneumonia that people hadn't seen before. So it was the first cluster of cases, and as we now know, it may go back a little earlier than that, but it pointed to this market as being the cause for that. And the Chinese were actually pretty efficient within a week of this appearing on ProMed and people beginning to get concerned about it. And it wasn't easy to get concerned because it's the New Year festivities, and so people were distracted by other things. China moved in and closed down that area of Wuhan and eventually the whole city. Now, through January, the cases began to rise, and by about the mid-month, we began to get the first deaths from the disease. And one thing became very worrying straight on, that there were plainly quite a high death rate amongst the cases of people being infected. And this seemed to be different from SARS, which had appeared about 16, 18 years earlier. There had been similarly symptoms. The symptoms of SARS and COVID are similar in some ways, but crucially different in others. But the death rate for SARS was never as high as this. And this worried people. Quite quickly, we began to see the disease spreading, although they managed a good job of slowing it down in Wuhan. It was in surrounding districts 3 weeks into January. 4 weeks into January, it spread throughout China. And as we go through into February, it's spreading out through China and into the rest of the world. And this is mainly people moving around and bringing the virus with them. And so once you get the end of February, early March, within China, which was still most of the world's cases, 2 months in, if you like, there were about 80,000 recorded cases and about 3,000 deaths, which is lower than we thought early on, but still high and still increasing. And indeed, once we go into March, we know it's already in Australia. It's arrived in Italy, where it's doing very well for a disease. There were quite a lot of people infected. We think this all came from one person from China who visited Italy and managed to trigger an outbreak there. It had spread to Japan, Iran, and it's being detected on the Western United States and also in Brazil. Mm. Now, let's step back a bit and say, is this an unusual event? Well, my colleague, Mark Woolhouse, along with a bunch of us, have been putting together data on this type of event. And this is, I think, the best chart, one that Mark put together for how often do we see new viral pathogens in humans? And he got the data back to the beginning of last century. And the key point to notice here, in the beginning of last century, we didn't know what viruses were. We knew people got sick. We couldn't find the particles because we didn't have powerful enough microscopes to look for them. But we knew that you could falsify postulates by taking some serum from people who were sick, passing it through a filter, and still being able to transmit it to people who got sick again. And eventually, once the technology improved, we were able to identify those things as viruses. The first one identified was actually a plant virus, tobacco mosaic virus. And the initial increases in the slope of this curve from about 1905 through until 1930, and then from 1930 through until 1950, are just improvements in our ability to detect viruses and see that they were associated with particular disease symptoms. Since around 1950, the curve has been pretty linear, where we discover of the order of 2 new viruses that's jumped across into humans from wildlife, 2 a year, 1 every 6 months. These are novel pathogens that we haven't seen before. Ones we've seen before, we may get 4 to 8 coming across every year. They try and establish, usually they die out, or we know enough about them to identify them and be able to control them. And we can put many of your favorite viruses on there. HIV is the one that caused the previous pandemic, picked up and identified in the early 1980s, but it may have jumped across as early as the late 1920s or late 1930s and circulated in populations in Africa, moving very slowly from one infected community to another. SARS, which is similar to COVID, came across just about 2000. Luckily, we caught it in time, and for one very good reason, it was relatively easy to control. Zika virus, which came across 2 years ago, or 3 years ago in 2017, was actually identified in Uganda in the 1960s. Zika is a village in Uganda, but it didn't spread from that village in a way that caused public health popular problems until it turned up in Brazil in the spring of 2017, when it started causing all sorts of problems, particularly with pregnant women who were producing babies with all sorts of growth anomalies. So this year we have COVID-19 that's jumped across. It's easily going to be as big a pandemic as HIV. It is now everywhere, and if you watch the news every morning, the mortality rates are not declining. They're still continuing to go up, particularly in the US. A lot of this is to do with how good a job we do in trying to control these populations, but pretending these pathogens are not there and praying for them to go away is arguably not the best way to deal with it. You can get an impression, and this is actually data from Spain, so I apologize, there are actually two graphs that are in Spanish. This just gives you for each year of the 20th century what the weekly mortality rates have been. And this thickest line is the curve for this year. You can see that it wasn't easily detectable from other viruses and other respiratory infections that are coming until we get about 10 weeks into the year, when we get this huge increase in mortality due to COVID in Spain, which, because of all the shutting down that you guys did, you managed to get a control on and knock it way back down into the region of similar to seasonal background mortality rate as you have from year to year. But that is still going to require lockdown to remain in place and released only slowly. All around the world, different things are happening. If you go to New York, although there's still denial from Washington that COVID is having any impact, these are cases in different blocks of New York City, and it's just increased massively throughout this big city. And the more we look at it, the dynamics of these pathogens are, they are always initially diseases in big cities because you have such a big population there. Slowly they move out into rural areas. And one of the things we've been discussing here this week, the people's assumption that it would be like influenza, and we deal with influenza every year. One of the things that instantly makes it not like influenza is we shut down all the transportation networks. So the spatial models for influenza that are based on all the airline flights and the train flights and the trucks and vehicles moving around aren't going to give a great representation of how this spreads spatially, because you've removed the primary forms of people moving around. But that also means the epidemic is going to go on much longer, because the only way you can get an epidemic is if people get infected, and if people are moving into the rural areas more slowly, it'll take longer to build up there. Now, some nations did a good job of responding really quickly. If you think back, it seems ages ago now, to China in February, they built a hospital in about 3 days. And indeed, by the middle of March, they had the first patient cured who'd been sick, been in hospital, and emerged cured from the disease. There were many, the weird thing about this disease is the longer you're infected for, the worse the symptoms seem to get. And even the last couple of days, we're seeing evidence of sort of brain damage in people from the UK who've been infected for a long time. And the people that I stay with in Panama, because I was down in Panama when this took off, one of them's been infected, and then plainly, it does horrible things to your lungs. Now, let's again step back a bit and think, what causes these pathogens to jump across? Well, about 10 years ago, a group of us in the US got together to pull information on essentially what is the dynamics of pathogens at that human-animal interface. Because to a good approximation, most novel infectious diseases of humans come from some interaction with an animal species. Historically, that's been interactions with domestic species. So if you look at all the major infectious diseases of humans that your kids get and now get vaccinated for, measles, mumps, chickenpox, those all arrived in humans as we domesticated animals and as we aggregated ourselves from small villages into bigger cities. That route through domestic livestock is still a likely way for humans to get infected if the livestock is interacting with wildlife in different ways. And that is one of the problems with these wet markets in China and other parts of Southeast Asia. If you bring wild animals into those markets and keep them at high density in cages in unhygienic conditions, then they will get stressed, and it's likely that respiratory infections they have may pass to domestic food that's for sale or domestic livestock that's for sale in those markets, particularly if that livestock is also live and only slaughtered in the market. But if we look at these diseases, it's quite remarkable that there are a number of hurdles they have to cross to be able to get into humans. There's obviously it's out there circulating in wildlife and groups of people trying to get an assessment of how many species are out there. This is a huge task because as far as we know, most wild species have somewhere between 5 and 20 viral pathogens, bacterial pathogens associated with them. So although we don't know how many other species we share the planet with, if that is somewhere, we know that the vertebrates are of the order of 500,000. So if you have 500,000 vertebrates, each with 10 to 20 pathogens, that's another 5 to 10 million viruses out there, a tiny proportion of which we've been exposed to. Now, when you look at the rate at which new viral species are discovered as people go out and sample bats or monkeys, then what you tend to find is once you've sampled of the order of 500 to 1,000 individuals of a species, you've found more than half of the viral pathogens that's going to be in that population. And that number is usually of the order of 60 to 80. So that actually says something interesting is going on. If there are 60 to 80 viruses circulating freely in these, and we've only seen 1 or 2 every year from all the species that are out there, that these things are challenging to get into humans all the time. And the majority of them, 99%, don't ever establish. You may get one person sick, but their immune system clears it. So it's really hard for them to go from this exposure and actual physical contact with humans to actually begin to take off. The next stage that happens is, if it gets in, does the person get sick enough to be able to transmit it to other people? And if they do, can they transmit it at a sufficient rate to make R naught, the number of people they infect, bigger than 1 or less than 1? And there's quite a lot of work looking at this. You can actually say what will happen then. Most of you will be familiar with this, but essentially, is the virus sufficiently transmissible to give you an epidemic? Oops, sorry. And will that epidemic produce immunity such that as you reduce the number of susceptibles and increase the number of resistant hosts, the infected individual's contact rate with susceptibles goes down to such a low level that R naught drops below 1 and the disease begins to die out? What we're seeing with COVID is that we can also do this by stopping contact between people, putting them in lockdown, getting them to wear masks. This will also slow the rate at which this curve goes up, and hopefully you can turn it over purely by behavior. Some countries were naive enough to believe you could turn it over by building up immunity, but it doesn't look as if we've built immunity up to sort of levels which are going to have to be at the 50 to 60% level where it will begin to reduce this down. The other hope is that we are able to develop a vaccine. And by vaccinating people, you massively increase the proportion in this blue curve, and that again breaks the chain of transmission. 2 things we need to know there is how long does immunity last for, and how efficient is the vaccine. And the one downside there is COVID, compared to something like measles, is quite a big chunky virus. It's like comparing an elephant to a mouse. And in general, the bigger the physical size of the virus, the shorter the immunity and the less efficacious the vaccine. So although people are optimistic, we might have a vaccine by the end of the year, you might have to take that vaccine every 6 months, and everybody on the planet might have to take it every 6 months, which would mean global vaccination manufacturing ability would have to increase by about a factor of 10 from what it is now. And so someone might make a nice profit eventually out of that, but it means a huge switch in the effort to make a vaccine, even if we get one that is effective. Natural immunity doesn't last for very long. You've got to vaccinate people multiple times. So what you're actually seeing, and obviously when this first started, there's almost, as the editors of Science and Nature tell you, there is an epidemic of people trying to estimate R naught. And obviously the problems with that are if you try and estimate R naught early on in the epidemic, you get a very fuzzy estimate of it because you haven't got very good data. As the disease progresses and you get more data, you're no longer estimating R naught because you're beyond the early stage of the epidemic. You're estimating just capital R at that time. But the more data you have gives you a better chance of being able to estimate what it is more accurately. So the better chances you have of being able to estimate it, the more likely you are to not get it published because so many other people will have published their wrong estimates ahead of you. So there's an interesting aspect like that. You then want to develop some structures that allow you to sort of predict into the future what is going to happen. And of course, you always hope you're going to be wrong because your main reason to use these is to look at how different types of control will send it on a different trajectory. So in many ways, you want to have models that are useful for testing out your control strategies to find out what's the most viable and economically possible, socially possible way to send the disease on a trajectory where R drops below 1 and the disease can be controlled. I don't think we're going to be able to eradicate this one. I think we're now with this one for the rest of our lives. So we are entering the COVID issue. As I said, there is an epidemic. This is just looking at SARS, Borrelia, which is a sort of older pathogen that jumped across West Nile virus. Each time this happens, the number of papers published goes through the roof. And notice that there's quite a few papers published, obviously, describing what's happening, and then an increasing epidemic of modeling papers. And that's very much what we're seeing. We've already had of the order of 40,000 papers published on COVID this year. It's surprised there's any trees left in the forest if they still printed the journals on paper. Now, where do these things come from? Well, there seem to be two main sources. The one I've hinted at before is the wildlife trade. This is a huge multinational industry. It employs many, many people, a tiny proportion of them legally, and it deals with of the order of 40 to 50,000 species of animals and plants each year. If we look for species that are the source of COVID, everything that the geneticists have done strongly points to bats as being the host and the natural reservoir hosts for COVID. It's not necessarily the case that a human that was infected by bats, though the bats are quite widely eaten throughout Southeast Asia. It may be that a captive live bat in a wet market somewhere was so stressed that it peed or pooped on some other animal, a raccoon dog or something like that, or some shrimps that people were eating, and case 0 was actually infected by eating something that wasn't a bat. But just to give you some idea of the magnitude of the wildlife trade, some have been putting this together. These are just data for the numbers of species passing through one country. We chose Singapore because it's a tiny country, so none of these species are from Singapore itself. Singapore effectively has no biodiversity. It has a few birds in the park in the middle of Singapore, but this is stuff imported and exported to Singapore throughout the world. And literally These are species from throughout the world. And when I look to these data, there are as many species from South America as there are from Southeast Asia, as many from Africa. And they are predominantly birds going into the pet trade, but carrying the risk of carrying avian influenza. There are quite a lot of mammals, quite a lot of which is associated with the fur trade, but also the traditional medicine trade. And then there are some plants, some reptiles, again, more associated with the traditional medicine trade. Two things dominate the patterns in the data you're seeing here. One is the increase in people following the rules of CITES and CITES. The Convention for International Trade in Endangered Species was set up in 1970. So these data reflect the organization being better at being able to detect trade, but also trade increasing as the global economy gets more international and more powerful. And you can see that it is the global economy, because once we get to 2008 and the global economy contracts, we see this contraction in the amount of animals in the trade, which begins to expand again later. As I say, you can divide it into different species: birds, mammals, plants, fish, corals. I should point out that the plants do not include any tropical timber. These are mainly sort of decorative plants for people's homes. The fish do not include any of the commercial fish that you go out and eat in restaurants. These are all little pet fish going into the pet trade. Now, one thing that I think helps clear up some misunderstanding, these are still these data from Singapore. These are the previous data are the numbers of species. You're looking at over 200 bird species, over 80 mammal species, 80 species of corals, etc. These are the actual numbers of individual transactions. And this is just the stuff going through Singapore that comes to the United States. And this is stuff going through Singapore that goes to China. So notice one key thing here is that, again, you see this rapid ratcheting up as CITES gets established. And then the amount of trade coming into the US is relatively constant, with only a tiny blip for 2008 slowing things down. If you look at what's going into China, you see the growth of China's economy and people being able to afford more exotic things as well as traditional medicines. But China's trade going through Singapore is always never gets to more than an order of magnitude less than the United States. So the US pointing the finger at China and saying, you're responsible for all this is egregiously wrong by a factor of 10. The US is the biggest importer of wildlife in the world. There are distortions in this because it does depend on reporting rates, but I don't think the reporting rates are more than a factor of 10 different. So the wildlife trade is one key thing that's causing lots of these problems. The other thing is tropical deforestation. Sometimes this works, sometimes it doesn't. Today it's not going to work. Okay, this is the Amazon, the world's biggest tropical forest. It is being chopped down at a very rapid rate, as is the forest of Central Africa. Those are the two biggest areas of remaining tropical forest on the planet. They have the largest diversity of species on the planet, and therefore they have the largest diversity of unknown viral pathogens on the planet. If we look at where Ebola, another emerging disease that's been causing huge problems in Central and West Africa over the last 20 years, where do the Ebola outbreaks occur? They always occur along the edge of the forest, where the forest is being chopped down and people are living in temporary logging camps and often eating local wildlife as food. So all the outbreaks of Ebola don't occur in the middle of the forest, they all occur around the edge. And just in more detail, both West Africa and Central Africa, Ebola is massively associated with recent land conversion. So what's happening here? Well, what's effectively happening is to say, if we look at the path of which these pathogens jump over, they tend to be circulating in the forest, in some forest-dwelling species. If humans go into the forest, they may become infected in the forest, and then take it outside and infect people who don't live in the forest. It may be as you chop down the forest, the animals come out and are able to infect you because they're running out of habitat and moving between different habitats. It may be that you could take the pathogen back to the forest, but there's this constant to and fro with very rarely life cycles that keep going outside the forest and inside the forest. Which means if we want to understand the emergence of these diseases, we've got to look at how this land use change pattern and changes in the patterns of human behavior lead to these pathogens moving across that boundary. So let's look at two specific examples. This is a place in Malaysia where I went to 20 years ago. That was because we had an outbreak in Malaysia of Nipah virus. And for a long time, we thought that Nipah and Hendra virus were the two scariest viruses out there that we should be worried about. And this actually is the farm where Nipah virus first appeared. It's heavily protected now. You can see there's no wire on the fence to protect the farm. And there are signs, both in Malaysian and English, around the farm that say, we accept no visitors in view of the outbreak of swine disease elsewhere. I mean, they're even pretending it's not there. But effectively, what's happening here is when this disease outbreak had occurred, it occurred in pigs and then spread from the pigs to the pig farmers. So as this was the index farm where it first appeared, they went in and destroyed all the pigs, and then they destroyed the farm to prevent it happening again. And this guy here, Dr. Zhu, is in fact the guy who identified the first patient, treated the first two patients who came down with Nipah virus. This is Peter Daszak, who's worked to set up groups of people to go out and survey for these samples, for these viruses in bats. And this is actually, Peter, this is one thing you never see for most diseases. This is patient 0. This is the first person to be infected with Nipah virus. He's the farmer who was working that pig farm when the pigs got infected. He got really sick, passed it on to his family, but luckily he recovered, but he still has very shaky hands and signs of sort of neuronal damage. Now, how did this happen? Well, again, as I said, what is happening in Malaysia is there is massive land use clearance, partly to collect, to build farms for pigs, because pigs are a major source of protein in Malaysia and other parts of Southeast Asia, particularly down here in Singapore. But those pigs, through the creation of these farms on the edge of the forest, become associated with bats, and particularly fruit bats. And how does this work? Well, this is a fruit bat, and this is a mango. And fruit bats love mangoes, but notice it's quite hard to fit that mango inside that fruit bat. So what fruit bats do is they tend to eat mouthfuls of mango, chew it, because they don't want to carry a lot of weight as a flying mammal. They get all the juice out of it, which is rich in proteins, rich in carbohydrates, and then spit everything else out until they get to the seed, when they also spit the seeds out. And when they do that, the seeds drop down into the places where the pigs are feeding. The pigs eat the seeds and the regurgitated bits of pith, and they get infected by whatever's circulating in the bats. Now, there are a whole bunch of interesting viruses in bats, but we almost never see a sick bat. All the bats that have been kept in zoos or occasionally resuscitated by people who do helping sick animals from the wild recover, no one has ever found cancer in a bat. All of which that suggests is that bats have immune systems that operate in subtle different ways from non-flying mammals. We worry that what that means is that any virus that circulates in a bat is used to living in a condition with a bat's immune system, which does a pretty good job of controlling it. Once that virus gets into humans, it replicates and goes into different tissues. It's almost as unhappy as you are when you're infected with it, because most of the time it's going to die out, as we've seen by those 99% of the viruses that don't establish. occasionally it will establish, but often it will manifest very strange pathology because it's in a completely different type of sets of tissues than it's used to being in. And with Cara Brook, we've been trying to look at that and look at the dynamics of viruses in cell culture from bat tissue versus mammal tissue, and they operate completely differently. So that should make us nervous about bats. And indeed, surveys that Peter Daszak and his colleagues at the EcoHealth Alliance have done show that if we look at the abundance of different wild mammal species and the number of viruses in them, then bats and small mammals and primates are the things we should be most worried about. There are other groups that we were previously worried about, things like goats, but we've already massively domesticated lots of those. So we've been exposed to many of the things that are all in the wild goat-like ancestors. It's really the world of the wild cattle-like things. We've been exposed to those. That's how we got measles. That's how we got brucellosis and other pathogens like that. So as I said, if we go and survey fruit bats in Malaysia for Nipah virus, we find it at very, very high prevalence. Often up to 80 to 90% of the fruit bats have Nipah virus. That's a really strong indication that it's not virulent at all, because with any pathogen, the higher the virulence, the lower the prevalence, because if you have a high virulence, it's going to kill you and not many of you are going to be infected any time. But having 80 to 90% of them infected suggests that it's circulating quite happily in the bats, and that anything that increases the contact rate between the bats and the pigs or the humans will increase transmission. And that's exactly what's been occurring if we dig a bit deeper. This is the pig density. This is work by a student, Juliet Pulliam, who worked with me back in the early parts of this century, looking at Nipah virus in Malaysia and then a little bit in Bangladesh. She showed that the pig density in Malaysia mapped very closely onto the mango density. Because when the pig industry expanded in Southeast Asia, as the markets from China and other places increased, okay, highly profitable to people to keep pigs. One thing you have to do with pigs is keep them cool because they overheat. So you can keep them cool by planting mango trees around where they're living. That supplies some free food for the pigs because the mangoes drop down from the trees in to feed the pigs. But it also attracts bats to feed on the mangoes, and they increase the rate at which mangoes fall down into the pigs. And those mangoes happen to be covered in saliva infected with Nipah virus, which introduces Nipah virus into the pig population. So you then get this crash once Nipah virus takes off. Now that crash is quite subtle. This gets more into the dynamics of this. If we look, and what Juliet did was to go back and look at when pigs started getting sick in and around Nipah, going to talking to people, making records, looking at their farm records. This is a mango pith that's been chewed by a bat. There were lots of little outbreaks of Nipah virus for about 2 years before the epidemic really took off. And that's quite unusual. But it tells us something that's both insightful for Nipah virus, and I think once we think about it a bit more deeply, important for COVID. What Juliet did was to model this using the relatively simple SER model, SEIR, susceptible, exposed, infected, recovered model for pigs. And what you find is when you get an initial infection, it's more than likely to die out. You get a burst of pigs that are infected, some of them die, you get rid of them, and then the disease dies out, but some of them have recovered. So when you get a second infection, it's not as big as the first infection. But because you've got so many resistant pigs in the population who recovered from the first outbreak, then the persistence time of the pathogen is considerably prolonged when you compare it with that initial epidemic. And in fact, when you generalize this parameter and turn it into a model, this immunological priming, as she called it, creating a partially immune population, leads to much longer persistence of an emerging pathogen than you get when it first jumps in, causes an epidemic, and dies out again. So the persistence time is very much a function of the proportion of individuals who are immune, which again points to things that are going to be quite scary with COVID. Although immunity is building up slowly, we don't know how long it lasts for, the more immunity there is out there, the longer the epidemic is going to go on for. Similarly, anything you do to slow down the epidemic, if it doesn't eradicate it, it increases persistence. And of course, understanding that requires a very interesting, delicate balance between, you don't get that if you look at deterministic models, you get some vague signature of it. When you start looking at stochastic models, the persistence time of something like COVID could be easily into the decades, even in small populations. And that's why I think we're entering the COVIDicene. Now, when we had this project, we actually had two fantastically bright women graduate students on it. The other one was Raina Plowright, who was working on Hendra virus in Australia. Nipah virus and Hendra virus are effectively sister species of viruses. This one is present in the fruit bats in Australia, and it caused a disease outbreak shortly after they had the Nipah outbreak in Malaysia. This time it occurred in horses, but for exactly the same reason. These were race horses that were kept when the racehorse season wasn't on in fields where mango trees were used for shade. There's a huge fruit bat community in Eastern Australia, and those fruit bats feed on the mangoes, drop them down, and the horses eat them. And a racehorse got infected. A vet came out to treat the racehorse, and he himself got infected from the racehorse and then died. So the symptoms that he was getting very closely resembled the symptoms we were seeing for Nipah virus in Malaysia. So people thought it was the same disease. Once they did the genetics, they realized they were closely related. And it looks as if these fruit bats do have a whole range of these viruses in them. The bad news for that is that these fruit bats range from Australia up to New Guinea, right the way around the edge of the Indian Ocean to Madagascar, and then in through East Africa to West Africa. So most of the fruit bats, which change family once you move into Africa, but all those fruit bats have these Nipah and Hendra type viruses in them. Now going and sampling the bats again and looking at how persistent the viruses were in the bat communities and putting some models together again, Raina was again able to show a very similar effect to that Julia had showed. If you look at them, this is taking populations of the same size with different levels of immunity at the beginning. If you introduce something like Hendra virus into a population that's never been exposed to it, you get a big epidemic, but it spreads through and that first epidemic curve dies out after about a year, 300 or 400 days. As the level of immunity in that population increases, because some people recover, the size of the epidemic, when you subsequently introduce it, is much smaller, but the persistence time goes up and lasts for much, much longer. So there's this trade-off between how much immunity is there in the population and how long the epidemic or the endemic, as it eventually becomes, lasts for. It gets smaller, but it goes on for longer, which means just as many people are going to get infected in the end. Now, eventually, we ended up, as I said, first of all, at NCEAS in Santa Barbara, and then in Sissing in Annapolis to try and say, could we make some models that look at this spillover effect and look at what happens as we begin to convert a patch of forest into a patch or bunch of patches of agriculture. There's this process, a few farmers move in, set up a little farm, the farmers have kids or other people here, it's a good place to be. So the agricultural area expands, the forest contracts, and essentially, if you look at the proportion of habitat converted, it goes from none converted at the beginning, it increases. the thing you're most concerned about from an epidemiological perspective is how long is the perimeter between the original forest and the converted land. And this may well become more of a fractal calculation, but essentially what's happening is that as the forest is converted, the perimeter across which disease transmission will occur will increase, so that the amount of disease transmission increases, peaks, and then decreases. Number of core species that have viruses you haven't seen before decrease as you remove their habitat. The number of matrix species, humans, cattle, sheep, dogs, increases. So the dynamics of any emerging pathogen lucky to get across that initial barrier will depend on the length of this perimeter and the relative abundance of core reservoir species and novel matrix species. So you can look at that dynamics, like, what is the relative rate of between versus within species transmission, and what's the relative abundance of the hosts. You can divide it between density-dependent transmission or frequency-dependent transmission and show that peak transmission is likely to occur when you have peak perimeter between core and a new habitat. That's the same whether it's density-dependent transmission or frequency-dependent transmission. If you do a stochastic version of that model, it becomes slightly more interesting in that your probability of getting an epidemic begins to peak out once you've converted 20-25% of the habitat. You've then got a relatively constant probability of getting an outbreak until you've got 75% of the habitat converted. But if you look at the relative size of the epidemic, it's not until you've converted a lot of the habitat that you get significant epidemics. And then you get a bimodal epidemic size. Once you get well beyond 50% and closer to sort of 70%, because this is when you've got a large population in the matrix. Although you're running out of reservoir species, if you do get an outbreak, it'll be substantially bigger than you would have got early on when you had less hosts to be infected. So you get that change in the size of epidemics. Early on, it's relatively small because you haven't got a large host population. At intermediate levels, you've got a very high probability of it happening, quite a large epidemic. But once you get out to 90%, you've got a low chance of having small epidemics because you're running out of reservoir hosts, but you've got a small but finite chance of having a huge epidemic because you've got so many new hosts out there. So what you then want to do is to sort of make risk maps of, well, where in the world, what is the viral diversity of the world in terms of where different host species are? And you can see here's the place we would be most worried about for bat diseases would be South America. There's a massive diversity of bat species in South America. But luckily, people don't eat bats in South America. They eat all sorts of other things, fish, and they eat monkeys. Monkeys is a worry because there are lots of monkeys and they do get eaten, but they have relatively low diversity of viruses. They've got other things. They also eat small mammals, and the diversity of small mammals and their viruses is quite high. And that just relates what I showed you earlier. So you could also look, slightly more subtle calculation is that as well as having a risk of diseases coming over, you can ask the question of, well, what role does biodiversity play in buffering us against these diseases? Because if you have lots of other species, maybe the disease just gets transmitted to them, they're not particularly good hosts and it dies out. So the second thing we sort of looked at is what sort of role might biodiversity play in buffering disease outbreaks? So you could look at the diversity of ecological communities in patches of different sizes, and this is aggregate data for lots of studies that conservation biologists have done for communities of mammals living in habitats of different sizes. Generally, as the habitat patches become smaller, the diversity of species living in them comes down, but often the abundance of those species that are left, their density goes up because they've lost all their competitors and they've lost all their predators. You can make relatively straightforward models of that, a sort of classic SIR model with subscripts for each of the different species, and look at how that all works. Luckily, you can also scale most of the birth and death parameters of the host species and the transmission rates of the pathogen, the density of the host, all to body size. And what you can show is, yes, if you make models of that, then but as you lose habitat, the host community collapses in the way you would expect from that you see in the data. The model communities collapse that way. You can also get a species area curve out, which corresponds to what you're seeing. But if you look at disease transmission, your risk of humans or domestic livestock being exposed to pathogens is very different depending on whether transmission is frequency dependent, which way is massively likely to be vector-borne diseases, versus density dependent, which would be more things like Ebola and COVID. So there, if you have very intact communities and you started to go into them, your risk of getting things like SARS and COVID at early stages of converting these habitats is significantly higher than later, because the abundance of hosts carrying them is declining as you make patches smaller. In contrast, the risk of getting a vector-borne or frequency-dependent transmissive disease, which is predominantly vector-borne, increases as we fragment up habitats, because the vectors that transmit those hosts, those pathogens, have got less hosts to choose from, and are therefore more likely to switch to feeding on humans and cows. As I say, there's now a whole industry of people looking at COVID and finding different ways to interpret data in ways that allow us to compare different countries. I think one of the most useful ways to compare it is to do this thing of normalizing the data around how many days has it been since the 100th case in a country. Because when you plot that out, the cumulative number of cases since the 100th case in days, you can plot on contours of doubling time. So you can see that those places early on, Hong Kong, Singapore, Japan, did a pretty good job by closing down the schools, quarantining, and having a very strong behavioral response, mask wearing. Their doubling time was consistently longer than a week. Places that were in denial, US, UK, had doubling times up every 2 days to 3 days. A very rapid rate of spread. Mercifully, we never got up to doubling times of every day, but some places got pretty close to that. There's been a huge push for doing different things, particularly keeping behavioral changes in place, but also getting people tested. A nice thing that my old friend Chris Dye, who's now working in the UK, but it was Chris who was very much responsible for setting up the global tuberculosis eradication program, and also was the WHO person in charge of Ebola and then Zika, points out that testing has this double-edged sword to it. One, there's two types of tests. There's tests you can do in the airways, looking for live virus, and that test is not an on-off switch. Your ability to detect live virus increases from when you were infected. It tends to peak just after you start showing symptoms. And then as you begin to recover, that test becomes less and less accurate because you've got less viruses, live virus in your system. So assuming that test is an on-off switch is spectacularly naive. You can get a positive test before you're actually showing symptoms. Obviously, when you're showing symptoms, there's a pretty good chance of it being correct, but we don't know how long you're infected for as you begin to recover. Similarly, the antibody test, which is where you're actually looking for an immunological response, evidence of an immunological response that remains active in your blood, doesn't really begin to pick up until after you've been pretty sick. And then the big thing we don't know with COVID is, what is this slope? How quickly does this come down, or does this persist? And how much can we enhance it by giving people vaccination, or even lifted up here before they ever get sick. Now, the crucial thing also about COVID that I said I didn't mention earlier, the thing that makes COVID very different from SARS is that you have a block of time when you can transmit it before you become infectious. That is completely different from SARS. SARS, you didn't show symptoms until you were infectious. So isolating people who began to show symptoms was a really easy way to control SARS. It was also a very easy way to control people with smallpox. If we look at that, just skip over that, very beautiful work that Christoph Braser did with Roy Anderson back at, again, beginning of the century. Was very fascinated with the R naught, with how quickly does the disease spread, and R. But the other vital parameter that's important for control is what are the proportion of infections that occur prior to symptoms or by asymptomatic infection. And with the 4 diseases they were looking at then, SARS, as I say, was easy to control because it doesn't have a very big R naught. And almost nobody gets infected. Effectively, nobody gets infected before symptoms appear. HIV, the pathogen that had caused the previous global pandemic, people could be infected for 10 years before they knew they had HIV and be transmitting during that time period. And that's what allowed HIV to spread massively around the world and still makes it really hard to control. Influenza is probably in the middle. Got a very high R naught, and lots of people get infected before the person who infected them knew they had symptoms. Smallpox, again, it's always regarded as a major medical success, but because the symptoms appear pretty much as you get infectious, it's relatively easy to control. And once we realized that when we began to get data from China about COVID, people can be transmitting before symptoms appear, which puts COVID down here. The R naught may be somewhere between initial estimates, people were saying 2 and 3. I suspect we're going to see it's much higher than that, more closer to 5. And this also may be more across to the right. A significant proportion of people seem to be able to transmit it before they know they're sick. And indeed, when we look at Italy, we're getting great data from there. Super papers by Marino Gatto and his team with Andrea Rinaldo. It looked very much in Italy, looking at this spread along the highway networks through Italy very quickly in the course of a month. Lots of this is due to people being infected with no symptoms, leaving thinking they're healthy, arriving finding they're sick, but having transmitted people and infected people along the route, creating these different individual epidemics, which get seeded in different regions and take off once isolation sets in, but having different regions at different stages with very weak connections between those regions is going to, again, enhance persistence. More and more people are doing great genetic things to give us, and we'll only really be able to do this in the early stages of the epidemic because now people are starting traveling again. It's going to get a much fuzzier picture, but essentially we know it started in China, made its way to Europe, but it radiated massively in Europe, and that took it into South America and back to Australia, even though Australia was started by China. China initiated in the Western US, Europe initiated the Eastern US, and the US has now got circulating things that will soon not be able to differentiate in this way. So we have had some successes in responding quickly. Wuhan did this great thing of by locking everything down, they pushed R naught below 1. Even as we're going into February, they thought they got rid of it, but it's come back. The US has not got the best people in charge of trying to get rid of it. What we've been trying to do over the last month, and hopefully this will appear in Science next week, is just to say, if we look at these different risks, the wildlife trade, spillover from the wildlife trade, monitoring the wildlife, what is the cost of doing these things? So these are costs in millions. Monitoring the wildlife trade more efficiently would cost us less than $500 million a year. At the moment, CITES, that the organization that's in charge of monitoring the wildlife trade, has an annual budget of $6 million. That is less than 10 seconds dealing on the New York Stock Exchange. It's expected to monitor the global trade in those 40 to 50,000 species with that. And it's explicitly said, our mandate is not to test for infectious diseases. But the budget to ramp them up, perhaps using the OIE, the organization based in Rome as part of FAO, that deals with spillover from livestock, would still be less than $500 million. That's a relative. Reducing deforestation to 40% of current levels would also be a good way to slow this down. It would also have additional carbon sequestration benefits that would more or less pay for itself. And just ending the wild meat trade in China, which is a big thing, you know, it's 20,000, it's $2 billion a year, but it's mainly luxury food for wealthy people. It's not subsistence food for people who need to eat. All of that gives you an estimate of the cost. We can compare them with the costs of the pandemic. So this is in billion, this moves towards trillions of dollars. Up until the beginning of June, the global GDP loss was of the order of $5 to $6 trillion. If you include in the mortality rates, trying to sort of look at that of people of different ages dying, that more than doubles that up to $12 trillion a year. The important comparison here, this is the cost of a pandemic. This little thickening of the line down here is all those costs on the previous page of doing things to stop it. But the relative benefits of putting more energy into stopping these things happen compared to the costs are just orders of magnitude. And in fact, you can see it there. The annual cost of preventing zoonotic diseases is somewhere between $22 and $30 billion spread across multiple nations. The damage from COVID so far is somewhere between 8.1 and $16 trillion. So it's a pretty no-brainer that taking some of those things and trying to do them is going to be a vital thing to do if we want to control the next one of these things to come along. And as I say, these things appear at roughly the same frequency as the US gets new presidents, or Spain gets a new president, or the UK gets a new prime minister. Partly suggesting that each president should be named after the pathogen that characterizes their reign as the increasing years. So I'm going to wind up there. I hope when you move away from your screens that you all wear face masks, because until we get a vaccine, protecting ourselves and protecting us infecting other people when we don't know we're infected is the key way to go. And of course, everything we see with this is trivial to compare to what happens when climate change comes along. So we still have lots of fun risks out there. Okay, I'm happy to take any questions.
Yeah, thank you very much. That was really a very nice overview and talk. So thank you very much. Yes, so we switch to the question session. Again, you can use the Zoom feature, raising your hand, or you can also switch on your microphones now and make a question. Maybe I start until we get some feedback. I would like to start the discussion. So I have one question that came immediately to my mind when you presented this. So recently I read about all these projects that try to find with genetic methods the viruses in different animal species. And there was a recent proposal about a very big project on that. I think they even thought about, $1 billion to screen most of the relevant animal species to get the sequences of viruses and then to screen them for risk, because from the genetic code you can do some analysis how risky it would be if you compare it to existing viruses if they would spill over. So in other words, there is clearly a kind of scientific competition between different approaches. And how would you see that kind of competition? Is it really a competition, or do these different approaches, well, maybe do give a better picture if they work together? Or what is your opinion about that?
No, in those calculations we did of what is the cost of prevention, we included in that $1 billion cost for the global virome project.
Yeah.
And I actually think that that is a hugely beneficial thing to do, because anybody collecting those data is obliged to put the genome sequences into a globally accessible database. It's not patented information in any way. So having, and you can't really keep live virus. So the best way to keep it is to have the genome sequences of everything. Then it's accessible to everybody else.
Yeah.
That stops that sort of proprietary competitive research. It just means people want to get competitive about getting more samples, which can only ultimately be beneficial. I am less optimistic that that will tell us much about virulence transmission. I think maybe in 20, 30 years time, we'll be able to go from gene sequence to being able to estimate useful characteristics of a virus, but it's a long way off. And assuming we can do it by comparison, we still have to have enough viruses which we know how they operate to be able to make that comparison. So that's a bit down the pike yet. What we do know is, having already done that for the things that are in horseshoe bats in China, we could quickly identify what COVID was and what the likely host was. So having that global virome museum or database allows you to identify a new human pathogen, and that allowed them to get ahead very quickly and develop tests for it. So you can develop the test for the live virus, that first part of those two curves, very quickly doing that. What you still can't do, but you again, having some sort of technologies, you can't develop the serology test from the viral genome. We don't know how viral genomes map onto serology tests. But if you knew the different major families of virus, what types of serology responses they do by doing experimental things in ferrets, I don't think you can do them possibly correctly in mice, but that would also get you ahead on being able to develop vaccine. But yeah, people say, oh, if we got the virus, we'll be able to develop a vaccine. That is complete bullshit. It's still a long way. We don't have that predictive power to go from genome to vaccine, but we would have a library that would allow us to do tests. And as I say, but most of it would be a sort of, that's why you need people like the WHO to administer something like that, so as there was a globally accessible database of virus genomes, and then you could find out where things are coming.
Yeah, I like that answer. So is there any other question by somebody? Heike?
Yes, thank you very much for this very inspiring and also shocking talk. So, and I would like to ask you about your last slide, the cartoon with climate change coming up. In one of your first slides, there was this overmortality pattern for Spain, and there it was quite clear that the heat wave in 2018 or so already caused a lot of death. Yeah. Not in the same magnitude, but already quite substantial. So what would happen if such a pandemic would come together with a heat wave? So for Europe, year 2018, together with COVID, would this be worse, or would that just add?
No, I mean, I think that's a fascinating question. We're seeing hints of this with, there was a very bad monsoon in India this year. So again, the monsoons push people in just as inside or push people together in different ways. What has that done to transmission, and how can we tease apart what was the enhanced transmission from the monsoon combined with having COVID there rather than you when you didn't have COVID. Once hurricane season starts in the US, it's going to hit the southern states where COVID levels are massively high at the moment. Now, what happens if Houston, where they're running out of hospital beds, gets hit by a hurricane in the next 2 or 3 weeks? Or Miami, where they're running out. So these things will interact with each other. So it is, mercifully, it hasn't been that hot a summer in Europe. Otherwise, it could have got much worse again.
And also linked to that, so could it be that climate change by itself would have an effect also on the disease transmission? So you showed the effect of land use change, and of course land use change enhances climate change, and climate change probably also influences land use change because other areas are needed for food production. So would you have an idea whether there could be a worsening effect of climate change?
It's a really interesting question. I think, again, when we think about the dynamics of diseases, I tend to sort of divide them into directly transmitted diseases, such as COVID and SARS, and vector-borne diseases. Now, we know vector-borne diseases are going to be much more susceptible to climate change because the developmental processes that go on in the vector tend to be poikilothermic, so they speed up as it gets warmer, and the life expectancies of the vectors changes as it gets warmer. So that would make me more worried about vector-borne diseases and climate change than I would be about directly transmitted diseases.
Yeah, but the bats could have another habitat then. Could imagine to have fruit bats in Europe.
When you start growing in mangoes in Europe, it will be a problem. And you have quite certainly be easy for some of the African fruit bats to eventually establish in Europe. It's not necessarily only the fruit bats, which are a completely different group of bats to the bats that you're used to seeing around towns and cities. But all bats have viruses in them, just as all small mammals have viruses. If climate change affects the behavior of those different mammal species in ways that increase their contact with humans, then you've got a problem on you.
I think it's a clear connection would be the loss of biodiversity and climate change. That is for sure an effect that will happen.
Well, again, you have this sort of double-edged sword with climate change reduces biodiversity,
as you showed,
and it's frequency dependent, and it's vector-borne diseases that you're worried about with climate change. Then that loss of biodiversity means that the vectors have got less choice of who to bite, so they switch to humans and domestic livestock.
Yeah, I would like to give the word to David. David?
You need to turn your microphone on.
Hello, David. Microphone.
Thank you for your talk. Very, very nice as usual. So enlightening. So several questions like come up to my mind, but one of those is related to the last part when you were talking about how much how more important would be like trying to prevent disease rather than by changing our habits, right? A lot, like for instance, eating less meat or eating not so, like rather than eating so much ham and so on, and you know, like the meat industry and so one thing we have learned about COVID-19 is that people can take like coordinated responses to the disease and lock down in their houses. But how much, so how probable do you think it is for people to change habits in a meaningful way, in a way that then we are preventing disease rather than having to fight against an emergent epidemics.
Yeah, no, I think that is a fascinating question of a sort of epiphenomena set of questions. One thing we've been trying to do is to set up a group of us that works with the economists and disease people, and partly because plainly pathogens have an effect on economics, as we're massively seeing with COVID, but also the economists are really a group that are interested in human behavioral decisions and what causes people to change their behavior in ways that affect their economics. But in this case, it's what causes people to change their behavior in ways that affect their disease susceptibility. And that's sort of the bottom line that all the behavioral economists say is it's so much easier to get people to change their behavior if you reward them than it is if you punish them. If you punish them, they'll react against it and keep doing the thing you don't want them to do. If they perceive there's some reward associated with it, they're much more likely to do it. So my dog that hasn't disturbed me yet, so the way we train our dogs is we only reward them if they do good things. We never punish them if they do bad things, but they just realize something's missing because they didn't get a reward or something like that. And my wife says she also trains me that way. So it works in several different dimensions. But in terms of COVID and other pathogens, how do you reward people for wearing masks? Other than telling them, yeah, you're not going to get sick. And I know it's been, well, I know in Barcelona yesterday, some bus driver got beaten up because he told people couldn't come on the bus without wearing, because they weren't wearing masks. In the US, it's really, really bad that it's become not wearing a mask is a political statement. And that's really stupid. It's trying to set up that behavioral change so there's some reward associated with it. And again, you then get into, well, what do people perceive as a reward? Or can they just do it as some mimicry of some of these sort of social people who get massively quoted on Twitter? If someone like that says it's really important to wear a mask, then lots more people of their followers would do it because they wear it by association. So it's trying to make people have a positive association with something can hugely change behavior.
Well, I think the problem is even worse. So yesterday I saw a report from Nigeria, and there a large proportion of the population doesn't believe the virus really exists. They think it's an invention by the government for some reasons. So, how do you change behavior if people even don't believe you? Because there is so much fake information around.
There's so much fake news that people don't believe it. But I mean, in many cases, I think, again, the US is going to be a curious case because lots of people have been in denial that the virus exists. Brazil also, the president says it doesn't exist, but now he's sick from it. Him getting sick and potentially very sick, hopefully that would change his mind a little. The British Prime Minister got very sick, and for a period probably slightly shorter than the immunity, he managed to change his behavior. But again, if they recover, in some cases they're going to have to get sick twice before they really believe it.
Is there any other question? Because I would like, I would have a question if there is no other question that interests me a lot. Okay, if nobody raises the hand, then I'm asking my question. It's a specific question about land use. You had this very interesting model which showed that the boundary between, let's say, the intact ecosystem and the transformed ecosystem plays a huge role. I mean, this means if you want to regulate that, it's again, of course, a behavioral thing. But I guess, for example, in Malaysia, where you showed the pig farms and the shading by the mango trees, I guess this was not a concentrated effort where some part of the land was converted, but maybe it was a very homogeneously distributed effort where you had little patches everywhere. And so you were destroying the forest in that way. Is there, I mean, I guess there is, your model would predict that if you would have a clearly more defined boundary, which you could only achieve, I guess, by regulatory measures. Oh, sorry. Sorry for that. So, which you could only achieve by regulatory measures. You know, is that something you would support? I mean, I guess it's relevant also for other questions in climate change.
No, I think, and again, it's Brazil is the interesting case there, where they managed to massively slow down rates of deforestation between about 2006 and 2015, and now it's massively picked up again, and it's all just change of will amongst the government to enforce it or encourage it.
Yeah.
Malaysia is also slightly, different from, the Amazon is this vast plain fed by the river. So it's a vast area that changes incredibly slowly as you go from the Atlantic into the Andes. Malaysia is this long, thin country that is limestone, which has this sort of massive limestone spine to it, which you can't farm, but it's surrounded by an alluvial plain. So the best agricultural land is along the edge of the coast until you hit that sort of limestone spine. The forest goes fine on the limestone, and it did fine as lowland forest, but it was easy to cut that forest down and convert it to pig farms. So you're always going to have this sort of shape along the edge of the limestone core of the country. There'll always be forest in the middle because you just couldn't farm it. But you could plant mangoes along the edge, it'll spread, so you'll have a sort of ribbon of mangoes around the edge of the forest, and then your pig farms along the edge of that. And that's roughly the more detailed structure of that map. And again, it depends who owns the land about how much it was converted or whether people wanted to keep it more as a lowland reserve.
Yeah, well, thank you very much. Yeah, I think this is really something we need to discuss, because that connects so many different areas as we have seen. Are there any other questions? Heike has a question, yeah.
It's me again. I wonder why the bats have this different kind of immune system.
No, that's a really, when we first, when we discovered that SARS was in bats, I was actually on the board of an organization called the Eco Health Alliance with Peter Dazat runs it now. And because it was originally a conservation organization that became interested in diseases, they said, we've got to write a piece to say that the answer to this isn't killing all the bats. The bats are not the problem. It's what humans are doing to them. So I wrote a little article and started saying, the most important thing we need to know is why bats are different. And then I had somebody wanted to come and work with me as a graduate student, picked up on that and said, can I work on that for my PhD project? So she started looking. I mean, we wrote one article that sort of says, why would bats be different? Well, once you initially think about how your immune system works, your immune system has a bunch of different, and then, one of the things I didn't talk about was trying to understand the mathematics of the dynamics of viral diseases within hosts is one of the things we've been looking at. Can you make models for the immune system and then introduce viruses into them, because they're just predator-prey communities where the immune system is the predators and the virus are the prey. In some cases, the virus is the predator attacking the immune system. So we started looking at it from that perspective to sort of say, how are there scaling properties of the immune system that would change as you go from bats to non-primate? Because bats live much, much longer. A 10-ounce bat can live for 20 years. A 10-ounce mouse is lucky to live for 10 months. So your immune system has to deal with living for a
Okay.
completely different length of time.
Okay.
The other thing is, if you're flying, most of your immune cells are produced either in your thymus, which in humans just atrophies once we get to 50, which is why you don't want to get sick much after you're 50, because you're not producing T cells anymore, or in your bone marrow, which is why you want to take calcium, because you want to keep your bones strong. Now, bats don't have a thymus. It just disappears because you don't want to have this heavy organ away from your center of gravity. And similarly, if you're flying, you have hollow bones. So you have nowhere to produce the B cells and nowhere to produce the T cells, which instantly gives you a different type of immune system. It turns out that bats, the one bone that's on the center of gravity, which is the pelvis in a bat and a bird, does have bone marrow, and that is massively reduced amount of bone marrow compared to even a mouse. But your B cells are produced there at a different rate, and your T cells are produced very early on, and then that atrophies after about a month, less than a month, or a couple of weeks. So the dynamics of the key components of the immune system are different. So we wanted to make models of that to compare bats with mice, and then mice with bigger things, to sort of see how having a viral infection is almost a chronic infection for a mouse, because it's going to last as long as you do, whereas having a viral infection, if you're a human, it's a tiny proportion of your life usually. So how do the scaling properties of the immune system change? And then what Kara also decided to do was to say, well, can we do the dynamics of the cell culture? So grow different types of pathogen in bat tissue, goat tissue, dog tissue, monkey tissue, and looking at that, the dynamics are very, very different, and the way the regulation of the immune system is very, very different in bats than it is in the other methods. So she's sort of continued to work on that. But I think that, I mean, we know there's tons and tons of viruses in the small mammals, and we know there's lots of viruses in the goats and in the primates. But in some ways, I would be less worried about the viruses from the things that are like us. They have a good chance of establishing, but also there'll be things that our immune system is used to dealing with. And it's dealing with the things that come from something that's been dealing with a different immune system that's completely different. And so I say, there are many, many ways of thinking about the immune system, but I think of it as the sort of Tom and Jerry cartoon, which I don't know what the Spanish word, but where the virus is the mouse, which gets into the house and does a small amount of damage, but the cat, which is the immune system, does most of the damage. And the more we look at COVID, it's the inflammatory response of the immune system to the virus that's causing massive damage in people who get infected. The virus seems to be clearing quite quickly, but it's creating this residual, really crampy, angry cat that's causing damage long after the virus is gone. And Andrea Graham, who's my colleague here, and she and I have been working at the Santa Fe Institute on these models for the immune system. The starting conditions of your inflammatory levels, or of the components of the immune system that are dealing with inflammation when you first get infected, are incredibly predictive of how you're going to do. And those change systematically with age, but there are also different lifestyles give you different relative levels of inflammatory immune particles. And if you've got the wrong combination of those, it's pretty predictive of what's going to happen to you. So again, there's lots to be looked at.
David, you had a question again?
No.
Oh, okay. Sorry, I saw your hand raised. Well, I think, yes, so we are coming to the end. I would like to thank you very much again. It was really very interesting. I really enjoyed it. And yeah, I hope we stay in contact.
All right, well, I'm happy. You all have my email, and I'm happy to chat with you all. Nice to see you all.
Yeah.
Enjoy your evening.
Yeah. Thank you. Bye.
Bye-bye.
Bye. Bye.